everyone. I am Professor Charu Dogra from Ramjas College, University of Delhi, and we'll be continuing with our series and coming almost to an end of biotechnology. Today we'll be discussing about the recombinant DNA in medicines part 2. In part 1 I talked about the generation and application of recombinant insulin, human insulin, and today I would be talking about the recombinant human growth hormone. So, the learning objectives for today's lecture would be to understand the application of recombinant DNA in medicine, and we will talk about the production of human growth hormone. The references used for educational purposes are basically the GLIC, the book GLIC about the molecular biotechnology principles and applications of recombinant DNA. There are a couple of other books as well, such as for molecular biology you can use Watson, and for particularly recombinant DNA technology that we have discussed in detail in our earlier lectures in biotechnology, you can also refer to T.A. Brown. So, the major question is when you see and observe around you, you see that everybody is not of the same height. So, how tall are you? You are among your generation taller, or you are shorter, or you are of a medium height. But one thing that is for sure is that everybody, all the human individuals are not of the same height. There is a lot of variations that you see. You also observe that some people are very, very tall according to their age group, and some people are really short. So, what governs this tallness? Now, if you remember, and you can also, refer to it, that in our genetics lectures we talked about that the inheritance of such traits such as height is governed by many genes. It is not governed by single genes. We talked about a detailed example of skin color, and similarly is the height. The height is governed by predominantly determined by genetics, which is 60% to 80%. With over 700 gene variants influencing the growth plate cartilage. So, you can refer to the polygenic inheritance lectures where we talk about that many genes additively, contributing a small amount give rise to the variations in the heights. And obviously, there would be different number of genes which are present in different individuals, so the height would also differ. Now, biochemically, the changes or the number of genes, how they express their function is by the key hormones. And one of the key hormone for determining the growth or the height of an individual, for example, is the human growth hormone. Then the sex hormones, the estrogen and testosterone, also influence during the, when they spurt during the time of puberty. And even the thyroid hormones play a key role in determining the height of an individual. Now, human growth hormone, which is also known as the somatotropin, this is the talk of the key factor of our today's talk. It is a 191 amino acid single chain polypeptide produced by the somatotropic cells within the anterior pituitary gland. Now, you always know that we have a endocrine system in our body that works through these hormones in influencing various expression of various characters in an individual. Now, the anterior pituitary gland is a major secretor of these particular hormones, which is again influenced by certain hormones released by the hypothalamus. So, this human growth hormone is released by the somatotropic cells within the anterior pituitary gland. Now, as you can see here that the amount of human growth hormone which is released by this pituitary located in our brain is basically governing the height, that if it is an appropriate amount then it will be an average height. If it is released in large amount you will have a large height and longer person or taller person, and if the amount is less then you will have a shorter person. So, this is biochemically regulated by the amount of human growth hormone present in a body. Now, human growth hormone is not released in the same amount throughout the life of an individual. It is more prominent during the childhood when you observe also that children grow in height quickly and then after a certain amount of time, which is generally at the puberty, that is the maximum time when the maximum human growth hormone is released. Then after that, as the age progresses, the human growth hormone decreases and therefore the height stabilizes. And in fact, various other, the growth of cells, the proliferation of cells also with time deteriorates, and even the aging occurs. So this human growth hormone, I just started off with an example of a height, but it is not just governing the height. It is also governing or influencing various metabolic pathways occurring inside the body or in the cell. So lot many things that it influences. But the key factor here we are talking about is the growth, which can be represented or observed as the height of an individual. Now physiologically, as you can see, that human growth hormone is released by the influence of another hormone, which is a human growth hormone releasing hormone that is released by the hypothalamus. So hypothalamus releases the human growth hormone, regulatory hormone, which is GHRH, which influences the anterior pituitary to release the human growth hormone. Now human growth hormone works by two different ways in a body or in a cell. One is a direct method and another is an indirect method. So in the direct method, the human growth hormone directly influences various, it is released in the blood and it goes into the various target organs, such as it affects the adipose tissue where it causes the lipolysis. It goes to the bone and the cartilage where it increases the absorption of various amino acids. The synthesis of proteins is increased, and therefore the growth occurs and the proliferation of cells occur. It also influences the liver for various activities. Then there is an indirect pathway where the human growth hormone affects the liver, or basically the release of the insulin growth factor 1, and this insulin growth factor in turn influences the various functions, again enhancing the functions of what we have talked about here. So the growth hormone can directly influence the target organs, or it can also work through the pathway of IGF-1, which is the insulin growth factor 1. Now this causes, basically again there are different hormones that play a role in the regulation of the release of this growth hormone that we will talk about. So this is the basic physiology. I am not going into too much of detail here because our main focus is the biotechnology aspect, but obviously you should know about the functioning of a particular thing that we are talking about in terms of biotechnology. So this is the basic physiology that we see. Now HGH production, as I said, is tightly regulated by several complex feedback mechanisms, and these mechanisms can act in response to stress, exercise, nutrition, sleep, and by itself the growth hormone only. So when we talk about that the hypothalamus is releasing the human growth hormone releasing hormone, and then anterior pituitary is releasing the growth hormone, it also releases certain other hormones as well, and along with these hormones, this mechanism is very tightly regulated, which is also associated with various external factors, that when somebody is in stress or when they are exercising or there is a proper nutrition or not, whether they are having ample amount of sleep or not, or the sleep cycle is disturbed, and when during the physiological development the growth hormone is released. So it's a very regulated manner. As you can see again in this flow chart, I have just tried to show you that what influences a positive regulation and what gives a negative regulation. So I have talked about three hormones here. One is the growth regulatory hormone, basically the human growth hormone. Another is the somatostatin, which is again released by the hypothalamus, and the third one is the ghrelin, which is released by the stomach, the gastrointestinal thing. So you can see that whenever there is a positive influence, so human growth hormone releasing hormone released by the hypothalamus is going to influence the anterior pituitary to release the human growth hormone. Now, this human growth hormone is going to, as we talked about, affect the fatty adipose tissue, or it is going to affect the liver also. Now, liver when releases this IGF-1, this is going to have a negative feedback loop. So, it is going to promote the production or act in accordance with the somatostatin to basically shut off the production of this thing. So, now this is basically, it creates a situation of hypoglycemia. Now, in addition, when the hypoglycemia is there, then the stomach is also going to release a hormone, which is basically going to tell about the hunger thing, and therefore it will influence the release of more growth hormone, because the metabolism needs to take place. So, this kind of a very tightly regulated manner occurs in the cell, in the body, so that the growth hormone is very appropriately maintained inside the body. And as I said, the increase or the decrease of the growth hormone is going to give rise to many disorders or many diseases, disorders basically, that is going to influence the growth of an individual. So, this one needs to understand the physiology as well as the hormonal regulation of this particular hormone to understand the entire system better. Now, the net effect of the regulatory mechanism produces a pulsatile release of HGH into circulation that varies hourly. So, as I said, it is not continuously released, but there is a rhythmic system of the release of HGH. In general, the HGH levels increases in the childhood, and therefore you observe a major increase in the height or general growth of an individual from the child to become an adult. There is a spike during the puberty, and in puberty other hormones are also released, the estrogen and the testosterone, which again influences the growth and height. Again, you would have observed this one fact that during puberty, when the other secondary sexual characteristics are developing, suddenly the children gain height also. So, suddenly they become taller during that 13, 14 years of age in boys, and that 12, 13 years of age in girls. So, at that time the human growth hormone also spikes, as well as other secondary hormones are, other hormones are released which influence the secondary sexual characteristics, and subsequently it decreases with the age. So, generally there is an increase in during the childhood until the adulthood is reached during the time of puberty, when it is maximally released, and subsequently with amount of time it decreases with age, slowing down the growth, slowing down the metabolism, and ultimately leading to the aging process which is there. The major aberrations in this HGH regulation may result in growth defects. Now, when we see the regulation of HGH hormone, we see that feedback mechanism is very tightly regulated, because any deviation in the amount of HGH produced is going to result in the growth defects. The HGH hypersecretion can lead to gigantism or acromegaly, and HGH deficiency can lead to growth deficit in children, the GH deficiency syndrome in adults. Now, when we talk about hypersecretion, we have written two things here. One is the gigantism and acromegaly. Both leads to an exponentially increase in the substantial increase in the height of a body, height of a person, or the size of a person. The difference is that gigantism occurs before the cartilage plates close. Therefore, there is a in general gigantism that is observed. The whole body is basically much taller and much broader, and it's a huge kind of people that we see. And acromegaly occurs when the cartilage plates, the bone plates basically have already closed, and after that there is a hypersecretion. So, again there can be a whole body enlargement as well, but then there is also certain parts of the body that become bigger. An HGH deficiency again can in children it would be much effective in case of children, because that is where the HGH is increasing and is required. So there will be a growth deficit, and it can also give symptoms in case of adults as well. Now I have put in certain figures of certain people here. You can see in the first leftmost corner top, he's a person called, he's named as Dharmendra Pratap Singh, and you can see that how tall he is. That's the case of gigantism. The entire body is very tall. Even on the right side, you can very well observe and recognize him. He's the great Khali, the WWE player. And again, he suffered from, and he recently in 2025 also, had a operation done, because whenever there is a tumor in pituitary, the HGH release will be, haywire. It will not be regulated much. So again, he has a huge and gigantical body. On the lower corner, the left hand corner, you can observe, that in circle. I don't know how many of you would be able to recognize. But that is a childhood picture of the great football player Messi, Lionel Messi. Now, not many people would be knowing, but Lionel Messi was also, suffering from this growth deficiency. And then, he was given this growth hormone supplements or injections basically, so that he could attain a reasonable height. Otherwise, he would not have grown more than 4 feet 7 inches, around that much only. So he's an example of the growth hormone deficiency. And again, in adults also, on the right hand side, you can see that growth hormone deficiency can lead to dwarfism. So, the man is really small in height, and even the children are not appropriately not appropriately, I would say, but not at an average whatever the height is among their own peers, etc. So these are some examples when the aberrations occur, when there is a too much of increase of growth hormone, or there is a deficiency of growth hormone, then gigantism or dwarfism or growth retardation can occur. Now HGH, so what happens is that obviously humans have to address to these disorders or these diseases, and what they started doing was they isolated the pituitary gland from the human cadavers, and they were used to treat kids who were unusually short in around 1963. So in 1960s, they used to isolate the pituitary gland and therefore take the growth hormone from there and inject it into the individuals who were unusually very short. Now in 1970s, although it goes on for like some 15-20 years, but by 1980 the use of cadaver tissue was restricted in the United States and Great Britain because of the possibility of transferring a syndrome, which is the Creutzfeldt-Jakob syndrome, and this caused Alzheimer and many different other diseases. So people were very scared. There's a mad cow disease, it's called as. So the symptoms just appear within 1 year of the injection. So people become very scared, or they were always intimidated that maybe they will develop these symptoms. So the infected cadavers that they took, it's a viral disease, so infected cadavers that they took and injected, then it was contaminated and caused this disease, this syndrome in the recipients, and therefore around 1980s it was basically banned. Then come the play of the biotechnology. So as we did in human insulin production, the biotechnology, as it started developing, it used the biology of the production of the growth hormone and developed a technique to increase the production or artificially, basically synthesize it in the lab, and therefore address these issues of short stature, dwarfism and other metabolic misfunctioning is also there. Recombinant HGH is a synthetic form of growth hormone used in clinical settings to treat severe growth hormone deficiency, short stature, and in fact even despite adequate hormone production. So it was addressed that there is certain diseases. We talked about the gigantism, acromegaly and growth deficiency, But again, there are certain disease which is called as idiopathic short stature. So, idiopathic short stature is when the people are really short, although there is a normal hormone production, but the functionality is not there. So, again this recombinant HGH would play a very important role. The first recombinant human growth hormone was produced by the company Genentech scientists in 1979 to around 1981, and they usually using the engineered in the E. coli, Escherichia coli bacteria. It was approved by the FDA in 1985 for public usage. Now, the procedure or the mechanism of the production of HGH was very similar to how they produce the insulin. So, you can refer to the human insulin as well. So, you can compare, we talked about in our previous lectures about that how these particular compounds were produced, molecules were. So, this was the first one that was released, it is protropin. So, protropin injections were released, which was injected into the people with short stature to restore their growth in the growth deficit people. Now, technically the production of HGH involved the HGH gene. Now, you can see that HGH gene is 191 amino acids, basically it is going to produce. So, what happens is in the 191 amino acid, there is another 4 amino acids which is on the top of it, and before that there is a signal sequencing molecule. Now, signal sequencing is basically when in a normal cell, wherever it is produced, the signal is going to make it be secrete outside of the body or out of the cell into the circulation, and therefore it will go into different. So, anterior pituitary, that somatotropin cells, trough cells are basically going to have this signal, the secretory signal, so that the human growth hormone can be secreted outside. Now this particular stretch of DNA needs to be cloned into the bacteria. So, they cloned that, but what they observed was that the secretory signal was not the E. coli was not able to cleave this signal sequence, and therefore, it was not able to release the growth hormone. Now, if it is not able to release the growth hormone, then obviously it will affect the production of the recombinant human growth hormone. So, because of this production of the recombinant human growth hormone, basically the difficulty would be there, and therefore what they observed was that there was a site of EcoR1 there. The EcoR1, when we cut this EcoR1, then basically it releases a 24 amino acid sequence which is up to this 191 amino acid part only. So, that 191 amino acid part is basically going to be cut off as well. So, they use that because when they were using this 24 amino acid part, the hormone was not able to be released from the E. coli. So, what they did was they removed this 24 amino acid part also by cutting it with EcoR1, and then they synthesized this artificially and put it together to make that 191 amino acid polypeptide part. So, that is basically how it is done. And then this gene was basically cloned in the lentiviral vectors or adenoviral vectors, and then in the expression vectors, so that the growth hormone can be expressed, and then they are cloned in the E. coli. So, this was one important modification that they did in the normal basic recombinant DNA technology. Usually what is done is that whatever that gene part is, that gene is basically that gene is basically cut off and introduced into the vectors and introduced into the host, so that the expression can be occurred, the cloning and the expression. But in this case, this presence of the signal sequence, that signal retention of that signal sequence in the protein was hindering its functionality as well as release into the external environment. So, they modified this procedure by cutting off the initial 24 amino acid part also, and you can see that they got this EcoR1 site there. So, that EcoR1 site is basically gave them an edge, and then they just cloned it, just like normal recombinant DNA technology we have talked about in detail. They cloned in the expression vector so that it can be expressed and produced. Now, there were certain limitations such as the loss of plasmids, development of antibiotic properties, lack of post-translational modifications. We have discussed in detail about these again when the insulin production was discussed. So, because the host is E. coli only, so the same limitations, similar limitations are basically arising in this case as well. The formation of inclusion bodies accumulation, improper folding, the proteolytic digestion and the poor secretion that makes the protein recovery difficult. So, there are a lot of limitations which are there. Now, human growth hormone, because when it is injected into the individuals, there are many side effects or risks associated with also, such as the joint muscle pain, the fluid retention, the carpal tunnel syndrome where the nerve associating with the carpals gets suppressed and therefore there is a lot of pain and tingling in the carpals, in the arms, in the hands. High blood sugar leading to the type 2 diabetes risk and injection site reactions. Long term effects could be related to acromegaly, that there is disproportionate expansion of certain parts of the body, cardiovascular diseases, cancer, gynecomastia, which is the development of breasts in males, thyroid issues, and worsened the scoliosis in children, the arch in the spinal cord which is there. So, there are many side effects and risks also associated with the production and injection of recombinant human growth hormone. The last point that I want to just state here is that you must have heard of HGH doping, which is used by the sports personalities, and it is a major issue in sports, which is used to increase the muscle mass, strength and aid recovery despite being banned by the World Anti-Doping agencies. So, many athletes, many sports persons are found guilty of using these growth hormones to increase the lean body mass. Although there are not many studies correlating it directly to the performance, but still it has been banned. And you should have an idea, and why I included this is because there always has to be a very ethical and responsible usage of all the technology that we create. So, the recombinant growth hormone or any recombinants are very useful for us, but it has to be used very responsibly and ethically so as to not lead to such cases such as of HGH doping. So, that would be all for now. Thank you.